Today we have three great talks. Today and tomorrow we have talks which are being sponsored and supported by the Human Brain Project, and specifically by a mini project called EpiSense, which is part of the Systems Neuroscience project within human brain. EpiSense is, it's been going for 2 years now. It came about as a result of an open call from within the Human Brain Project for new projects that would connect the that particularly wants to take that approach with respect to understanding the fundamental capacity for mammalian cognition, which is, as we heard from Medvard yesterday, to understand and find our way around in the world, but also in humans to be able to recall the past and also to make sense of the future, because it turns out that similar mechanisms underlie those two things. And I think actually it's a central part of the human condition, and one of the things that makes us different from other animals, that we do have a strong sense of our personal past and our history, and we use that to make decisions in the here and now and to plan for the future. And Cyriel Pennartz, who's going to give our first talk, is indeed the leader of Sub-Project 3 in the Human Brain Project, which makes him a powerful and influential person. It was his idea to put together this mini project, and he was fortunate that he invited me to be a partner. We'll also hear from Neil Burgess, who is actually, he's in EpiSense, but he's also in another part of Human Brain Project, SP4, which I think is more the theoretical neuroscience. Is that right, Neil? So we'll hear from a number of Human Brain Project speakers today and tomorrow, and we hope to garner the feedback of people at the school about the work that we're doing, and to find out from you what ideas you have. So please come forth with your questions. As Paul says, one brain, one question, and I think there are people we haven't heard from yet. So the final thing I say about Cyriel is that he was at the first ever Barcelona Cognition, Brain and Technology Summer School. So he's a veteran, and it's great to hear from you on our 10th anniversary meeting. Cyriel.
Thanks a lot. Tony, is this on? This thing? Yeah? Okay, yeah. Thanks for the invitation. It's a pleasure to be back. It's always been such a great mix here of speakers that talk about technology, cognition, and brain research. So, one feels like a fish here and in the water. So I'll try to do something impossible for you today, which is to connect the first thread of the summer school on consciousness with the second thread more on memory, spatial navigation, and so on. So I'm sure to be bound to fail, but I hope to make clear how several connections could work in that respect. Here, starting from the picture of Amsterdam, you might be used to coffee shops over there and know our city as such, red light district and so on. But did I do that? Here we go. Yeah. That's a lot better. Now this. Okay, there we go.
Can the man drink his coffee in peace here?
Well, not you, that's for sure. Whereas these arrows are actually not pointing to coffee shops, but to places where philosophers lived in the past, such as René Descartes here on Calver Street, and Spinoza here in the Houtgracht, and on red we have John Locke. It's kind of interesting in the context of the Brexit that he lived in Holland as a refugee from England. So that was possible at the time.
So you're saying you're going to adopt Tony now?
Well, maybe depending on how bad the Brexit will be, but you're certainly welcome. So yeah, today is first a bit about consciousness hallmarks and functions, the way I see it from a neurobiology perspective. That will continue to lead us to predictive coding, modeling, where we try to make world models basically based on primary sensory inputs that try to make sense and predict what's going on in the world and drive it upwards to higher structures in multi-layer networks. Then we'll turn to physiology, neurophysiology of multisensory interactions, and visit the perirhinal cortex and hippocampus, where we specifically revisit the question of yesterday that Rizzolatti touched upon. The question in this case whether the hippocampus could code more than self-parameters and also provide information about other beings, perhaps by way of mirror place cells. So first, when we talk about consciousness, it's a highly complicated topic. It's already quite difficult to define consciousness. It is what we lose when we fall asleep and what we regain when we wake up. But as such, many of these easy definitions resort to circularity. What is waking up? What exactly happens? But we do associate it with having experiences, with having qualitative experiences that are quite different from unconscious processing. So it's going to be difficult to define a consciousness head-on, as it were, from a primary definition, but we can look at hallmarks. We can see what kind of processes we associate with it and how far we could peel away brain's processes before consciousness is lost. So let's see, often consciousness has been related to memory. The remembered present we remember from Edelman, for instance. But here, by itself, by lesions, we can also note that patients like HM, etc., have shown us that consciousness does survive the removal of large sections of our declarative memory, even though memory can enrich conscious experience by way of providing information about the past and allow us to live ahead, to prospect into the future. Emotions has been very often associated with consciousness also, for instance, by Damasio, very much linking it to bodily sensations and the feedback of that on the brain. However, we also have long histories of lobotomies, etc., operations by which emotions were blunted, and still one would see the survival of basic sensory awareness and consciousness in those cases, even though, again, emotions might be important, or motivational drives could be very important in steering the genesis of consciousness. Metricity has been very prominent recently in debates on embodied cognition. There are people that, like O'Regan and Noe, try to explain even qualitative experience in terms of sensory motor contingencies by the rules or laws that would govern how motor changes lead to different sensory feedback, and how it's natural to see the genesis of color sensations from that. That makes a lot of ecological sense. However, we do have a lot of neurological cases where not only paralysis occurs, but also locked-in syndrome, and even patients born without limbs, like in Amelia, patients who do have sensations in their limbs which they never had. So that is a curious case of having conscious sensations in the absence of sensory motor contingency loops. So that at least would drive me to think that maybe on the long term, all this, of course, this motor interaction with the world is important, but not on an acute scale. And even Amelia or congenital blindness, for instance, would argue that such long-term input in one modality might not even be necessary to sustain consciousness. Language, often mentioned as a higher order phenomenon. However, we do have to think of not only aphasia patients who remain conscious, but also evolutionary conditions under which humans or hominoids had no language. At least there were no clear indications for language as such. but it's very hard to think of them as unconscious beings, and same for chimps and other animals. We do have probably then as core, the way I see it, perception and the more internally related forms of imagery and dreaming as being essential for consciousness.
Cyriel, wait, for these definitions, so you try to spell out what you call necessities for consciousness, right? And then in some sense we have the whole of psychology now on the slide.
Yep.
So can't you be more specific than that? I mean, because in some sense also HM, it was not that he wasn't conscious, just he didn't have declarative memory anymore. As an example, right? Yeah. If I have no language, it doesn't mean I'm not conscious, right? So what are we going to do with this list now? Are these benchmarks? Do we have to explain all of these things together to get an idea of consciousness, or?
No. This slide sort of explains this part. What can you peel away before it's lost? And the claim here I would make is that you can peel away a lot, in a strict sense. But not so much perception, imagery, and dreaming.
So you say you can lose those things, but not the last two.
Yeah. Whereas, strictly, yeah. So you can peel away a lot, even though the other factors could all enrich consciousness.
You talk about phenomenal consciousness, like experience, or you talk about, let's say, the information processing sense of consciousness, like access consciousness in the sort of block distinction.
Yeah, those are very important distinctions. I mean here phenomenal consciousness. Yeah. Access consciousness, difficult topic. I think it mainly has to do with logistics, making contents available for actions, working memory, etc. All very important topics. I hadn't planned to talk about it. Hallmarks of consciousness, so what would characterize phenomenal experience. The way I see it could be summarized as follows. So we mainly talk about perception here and derived more internally driven forms of imagery and dreaming, where in perception or imagery and dreaming, qualitative richness is a very distinct feature, the subjective aspects of experience, which seem not only confined the way we have experience to vision, for instance, as a dominant modality. All these submodalities we can distinguish in here, such as direction, color, kind of implied motion, etc., form. But it's also having to do with having a certain place and being in a context in what we experience. That is, we don't view this picture purely in some kind of abstract space, but we are there. We are, for instance, in the church in Rome, where this picture of Caravaggio is situated. So we have our vision accompanied by bodily sensations, proprioceptions, noting where we are in space, how our bodies are positioned, what we smell, and so on. So it's a kind of immersiveness that is multimodal in its nature. With that comes a sense of integration, because we do merge sounds, for instance, with visual lip movements, as in the case of the ventriloquist. So we tend to allocate different signals to the same sources. There's a certain dynamics and stability to conscious experience, in the sense that we cannot arbitrarily switch between the back and the forth of this Necker cube at arbitrarily high speeds. We do have a certain sluggishness in that sense, but there's also dynamics. We can renew or change experience. First-person perspective is certainly a usual hallmark, not perhaps necessary per se, but having this Escher-like sensation of viewing one object from multiple perspectives is artistic, but unusual in that respect. And interpretation, inference and perception would be also hallmarks, I think. So that there's not only purely sensation of such a duck-like shape, but also an interpretation of a semantic labeling of this as a duck or a rabbit, where we also can do a kind of semantic switching where the sensory input remains the same. And this vegetable picture refers to intentionality, so that the fact that we don't need to have vegetables in our brains to think about vegetables, and neuronal activities must be somehow generating a projection that those vegetables are elsewhere, neurons aren't.
Sorry, would you agree that these criteria also hold for subconscious perception? Like if you look at these tasks, like these typical subliminal perception tasks, right? You use words or faces to bias subsequent perception.
Mm-hmm.
Then in those cases, all of this would also hold, right?
As a sub-process, perhaps yes. For instance, dynamics, stability, integration can occur at a subconscious level, but I think what subconscious process would be lacking is the overall multimodal integration. That is, of course, proprioception could be integrated still with unconscious vision in the way we control our body movements, but I don't think it leads to the kind of, let's say, grand survey of the situation that we have in consciousness.
You have to draw the threshold somewhere.
Yeah.
Then
yeah.
it's up to you now to define that transition area where you say, well, it cannot be more than 2 modalities or something like this, or it cannot be more than X bits.
Yeah,
or
yeah. This is also an empirical method to sort out. There are certain, let's say, multisensory integration processes that will go on during anesthesia, but not all of it. Okay, those are very good additions. We come to the thorny topic of biological function of consciousness, which relates to this. And here, I think an important distinction is between what our brain can handle unconsciously, so to say, what we can do away with without invoking the whole business of cortex, thalamus, and the whole ball game of consciousness. And the proposition is that when we deal with single stimuli, in the way we act, like this tadpole on a shadow approaching on the water, it's easy not to invoke consciousness. You have to generate a reflex response and duck away, a stimulus response behavior. However, if we are in a situation that requires comprehensive decision making, such as this soccer player over here, then one has to take into account a lot of factors at the same time in that process. So it's not sufficient to see the ball and react to it, just kick it. You need to know, am I defending that goal? Is it my goal or is it the opponent's goal? How am I positioned in this? Where is the opponent? The color of the field, the whole fact that you play a game there matters. So it is much more of a multifactorial situational process where indeed I would argue that a multimodal survey of the situation is what you need to support this comprehensive decision making. And there we have an intimate link to goal-directed behavior, which is not simply behavior directed at a goal, but I mean it in the sense that Tony Dickinson, for instance, from Cambridge, meant it. That is behavior that is relying on a representation of a goal, on planning. So it requires a plan, but also knowledge of the environment. Let's say to have it all at once available in a very quick and dirty way, perhaps. So here also retrieval of information from memory comes into play, that you need to support this complex decision making. Hence the close interaction of consciousness with declarative memory. Many neuroscientists don't like to talk about consciousness and they sort of try to avoid it or label it as irrelevant. However, when it comes to defining declarative memory, they do rely on consciousness because it is defined as the conscious recollection. What you consciously recollect is what defines declarative as opposed to procedural memory. So it does come into play and it's hard to avoid. So here this slide also emphasizes how action doesn't need to be purely driven by all consciousness, but there's also the habit system with which there is interaction going on, as we also showed with, for instance, Giovanni Pezzulo in simulations. So it is having to do with prospective deliberate behavior, where, let's say, the planning very much also relies on memory and what you have at hand. That's the question, yeah.
Yeah, so sorry, I don't have the microphone. You say that this comprehensive decision-making takes into account the whole environment and body state, but sure, I mean, it's a bullet point, it's not a...
Yeah, it's all very short, yeah.
I was just wondering, I mean, I guess consciousness, there's this idea of the unified scene, And so there must be a lot of selectivity. I can't remember who.
It's on already.
Okay, I can't remember who said. Is on?
The light is on. It's red, off. Green, off.
It's on.
Yeah. Unified scene, you were saying.
Yeah.
Yeah.
So sorry. The point was just that you have a lot of more or less subconscious simulations running in your head, right? Of all kinds of events that could happen, conversations you might be having. And I guess consciousness is also about selecting between all of those possible, all of those processes and which ones come to mind, right? So it's a very strong sort of selective conflict resolution in some way.
Right. Yeah, that's why I'm also not saying what we experience is purely a sort of summary statistics, because we can focus on details, whereas we not necessarily lose the overall context in that way. And yeah, the process of selection is there in that I think one needs to focus on the relevant elements for decision making, not everything else. We often do have, of course, the idea that we see a whole scene, but if you really test subjects on it, like done by a Sid Cui there and so on, it appears that in the periphery we just have a very coarse knowledge of what's going on. But with that, I would also like to keep attention a little bit apart, because there are quite a bit of psychological studies also showing that there can be consciousness without directed attention of something. That is, we can focus on something but not be totally unaware of other things going on. And there could also be attention also in simulations without consciousness, I think so. I think I agree with you. Yeah.
If we now talk about action planning and action selection, in case of the football players, for instance, you're saying that the conscious state, or what you call representation, is influencing the real-time behavior of that soccer player. You see it as a factor that is driving the real-time performance of that agent. You follow the arrows, right?
Yeah, indirectly through this stage. So it's not the case that if there is action, there must be consciousness preceding that, or conscious deliberation. But I would maintain that, well, of course, we can also be conscious without deliberation and planning, just by mind wandering. But if we look at the function, which is the topic of the slide, then I would say that this is a very important arrow. That is, for deliberation and planning, you can get a lot done with purely, let's say, subconscious computation. But if we would lack consciousness, that is, we would lack the overall world survey that we have, then you would be in a bad situation, because then you don't have this survey at hand. It's not there, so to say. So that would be, I think, a big handicap.
But on that arrow, from conscious to deliberation, you would have the limit factor, right? So we would have some sort of delay. So you're going to deal with it later?
No, I would just say that the factors that Libet also implies, like motor readiness and etc., the readiness potential, are additional factors that play into decision making and action. So that could also explain why, we don't have a pure synchrony between these processes.
All right, but then in this model, so you're going to explain to us how the conscious representation gives you the whole integrated picture of the task that is driving the deliberation.
No, that's not what I'm going to explain. No. This is mainly to put on the map consciousness versus declarative memory, planning and deliberation and action. So mainly due to time constraints, I would like to talk about it more, but that's going to be quite hard. I do want to address a little bit how the brain could come to create this rich multimodal survey that I talked about, how perceptual experience does come to be rich, which is, of course, a great philosophical problem, qualia, etc. There's no shortage in itself of sensory feature detectors. We have in each modality lots of, let's say, cutaneous receptors, etc. So in that sense, there's a great richness of sensory inputs. The problem is more that from the brain's viewpoint, these sensory inputs are unlabeled, so to say. They arrive in the brain from the periphery, all by the same kind of action potentials everywhere entering the brain. And there, the sensory physiologists would say, well, okay, there the brain takes over. It makes sense and generates cutaneous sensations from these inputs. But also from the neural network viewpoint, basically we're dealing with numerical operations, computations, where the inputs and outputs are unlabeled. It's, as it were, Chinese. There's no, it's all in the eye of the beholder what the networks are processing. So that's a big problem. In that sense, I'm also not saying that that neural network approaches would solve the symbol grounding problem. They actually don't if you compare it to physiology. What we can do is look at diversity of unidentified sensory inputs and how the brain constructs world models from that. Here, inspiration comes from Helmholtz, for instance, who also talks about inferential processes and how representations of sensory qualities could be included as quick, functional, and distinct markers of what's going on. Like I said, we do have a problem here in that the brain lives in a kind of Chinese room, as compared to Cyriel's Chinese room, where the Chinese symbols could be also replaced by cuneiform symbols, namely spikes that are arriving. There's nothing in here that would say, well, you're now in the visual cortex, so the spikes become visual information. That's not an intrinsic given fact, so the brain will have to deal with it. What we can do is relate those distinct sensory inputs with other inputs and thereby compute statistics by which particular sensory inputs also become unique by the relationships they carry with other sensory input streams. For instance, in these pups, ferret pups, once the pups open their eyes, visual input could be related to tactile input, to audition, to vestibular sensations, etc., by which body maps can be built, by which a vision gains a particular identity also as being decorrelated from other senses. So in that sense, there's something to gain from looking at the interactions between the sensory modalities amongst each other. So some while ago, I proposed to also consider not only input specificity in, let's say, the uniqueness of retinal input as being important for vision, but also relating visual properties to other nodes in the overall multimodal network. So these would be not anatomical nodes, but rather perceptual nodes, where for humans, for instance, we would note under particular motor conditions, high correlations from vision to audition, proprioception, but less to taste and olfaction and so on. The kind of anatomical scheme it leads to is to actually not emphasize the visual nature of visual cortex, for instance, but rather say, well, there's an area here that receives a dense LGN retinally derived input. That area is going to interact with a lot of other areas that receive their own unique inputs, and that in the process of interpretation and interaction, something like vision arises, but that's not purely a matter of V1 or even V4 higher areas. It is going to be a much bigger phenomenon than the classical visual areas per se. So that does make it complicated. I cannot go into all the kind of predictions and complications in there, but I will address a little bit in the rest of the talk. World representations, how that could arise, models of causes of our sensory inputs, and present some physiological data on whether sensory cortical areas are indeed uniquely unimodal or could also have a multisensory component.
I don't really understand this slide. Because you have a bunch of boxes or spheres with lots of arrows between them.
Yeah, you love arrows, right?
Yeah, and then it's sort of you seem to be saying like, oh, okay, these guys do all things together, it's all sensory. But what's this telling me? Are you trying to say that vision is central to all of this? Is that the point? That's why you put vision in the middle?
No, it might be for humans, but for any organism it could be different. Even for some humans it could be different. The point is more to say, well, vision will not be a phenomenon that is going to be enclosed in a particular restricted portion of the cortex, like V1 or V4, etc. But rather is something where the areas that receive visual input are going to interact with other modalities, but also memory and motor domains, to in the end determine what we experience as vision. So it's, in other words, a network phenomenon, and not so much.
Vision is intrinsically multimodal.
Basically, yes. Yeah.
Okay.
Yeah.
But why
Yeah.
is there no action then? Because earlier you were talking about Kevin O'Regan and Noe, and you indicated that action was relevant in that, in the story of consciousness,
Mm-hmm.
I thought. But now you left it out.
Well, no, because action is in there as proprioception. It's the way we notice motor actions by sensory feedback. On the motor efference copy would be very important also to inform the sensory modalities of what's going on in terms of motor commands. So that would also distinguish active movement versus passive movement. That in the story fits in if we distinguish, that correlation between vision and proprioception, for instance, will depend on whether you're closing your eyes or moving your eyes, for instance, which is crucial. Okay, well, sorry to be so brief about it. Happy to continue in the discussion. I do want to touch upon one modeling approach, which is not comprehensive yet. We just started it in the context of HBP work also. And that relates to predictive coding. It's basically a way to implement the scheme of making a world model and trying to predict what's going on, even though we have no direct images from the world, but merely have to rely on action potentials, basically, that are coming in. Some inspiration, but in a limited way, comes from artificial multi-layer generative models, where, for instance, networks learn a joint distribution of digit images, concrete, let's say, sensory inputs, and digit labels or classes. And these networks, like designed by Hinton, are very good at indeed grouping different instances of zeros together into the class of zeros. And they can have a capacity also to classify unknown new test images correctly in that sense, so they can generalize. A generative model will have the capacity also to not only classify sensory data, but also to generate sensory data from class labels. So you would say, give me a 5, and the network would be able to come up with some kind of example of a 5 that maybe it never saw before. So in that sense, it might be a little bit compared to imagery or fantasizing based on semantic cues. But yeah, these networks do often still have, especially the deep learning variants, rely on backpropagation learning and other artificial principles. So one important question is whether we can replace those. We do have beautiful images from some of these generative models, like here in Guian, showing all kinds of examples of volcanoes, fictive volcanoes that the network comes up with as images that would optimally drive particular units in such networks. Or look at these beautiful artificial birds that are being created by the network. It's quite remarkable what they can do. But here we are interested in the neurobiology. So one question is, do multi-layer structures actually exist? And much has been said about multi-layer hierarchies, such as in the visual cortex, here rendered by Velleman and Van Essen. It runs up from P1 to the hippocampus and the entorhinal systems that you heard about. The sensory cortex to hippocampus will have this roughly pyramid-like shape with both upward and downward projections. Of course, the real anatomical schemes are way more complicated, but it's to convey the idea. And we do see the emergence of, let's say, higher up, bigger receptive fields, more invariant properties, selectivity for more complex feature combinations like faces, etc. We will pay attention most here to V1 type of properties, like sensitivity to orientations, to gratings, to oriented edges, and so on. But in the end, the genesis of these higher level features is going to be important. And like I said, predictive coding here is very much directed at modeling hidden causes of sensory inputs. One would project images into the network, apply them. The images would be processed by higher layers. The task of the network is basically to generate an optimal prediction, which is fed back to the lower layers. And what remains after this kind of subtraction over here, that is the discrepancy between predictions and actual inputs, is propagated forward as an error signal. So then you arrive at this middle layer, which has a higher order, a predictive representation, and so you would go on. But the problem has been that the existing models are only shallow networks, that is 1 or 2 layers, depending a bit on how you define it. And they were trained on image patches, that is not whole scenes or whole images, but rather small parts of it. But this has been done successfully, for instance, by Rao and Ballard, but also De Jaan and Hinton have designed their Helmholtz machine, not accidentally named after Helmholtz. And so we wanted to add more layers, make the network scalable, make a real deep predictive coding network, and then see what kind of representations will arise at higher layers. And it has to be plausible in the sense of learning rules that are compliant with neurobiology. We'd like, of course, to have V1-like properties in the lower layers. The aim is also to extend it into these multisensory domains and see what happens, but we're not there yet. We're just at the very beginnings. Shirin Dora is a postdoc in the lab who has started on this work. We do have some initial picture here where you see how indeed reconstruction errors are going down upon training. This is on a, for instance, a CFAR image data set that has to be learned, and you do see that a 3-layered network in this case is able to learn this. So the feed-forward error in this case is minimized, and that is good news for the learning. It operates on the whole image as input, but as yet we're working now to extend this. A 5-layer network has succeeded, so it has become really a deep network. And we do find already at the level of the lowest and second layers, these filters, or let's say optimal driving patterns for the units up there, which do have orientations. Let's say this dark to light edge has a particular orientation that would suggest orientation selectivity, but it's sometimes also more complicated structures like these dark bars or light bars. So in that preliminary sense, it's going in the right direction. We'd also say we don't have a natural consequence of developing high-level inferotemporal-like shape representations, for instance, or classes of objects, semantic categories. That is not a given. It doesn't seem to be arising naturally if you train networks like this. And we're just currently sorting out how that could be done.
Can you say something about the specific physiological benchmarks that you think such a model must satisfy?
Mm-hmm.
Like also when you start to scale now towards IT, at all these different levels that you're going to model, what are the specific physiological benchmarks that you think you must satisfy to make it, let's say, a plausible model?
Right, yeah. Yeah, so on the one hand, there are the postulates that learning rules, for instance, must be at least compatible with the neurobiology, preferably Hebbian, LTP, LTD-like, or STDP compatible spike timing dependent plasticity. Right now, what must be satisfied would be the low-level V1-like properties, such as I show here, so orientation selectivity and perhaps some more complicated ways to do it. Of course, we want to go on and also see how higher order representations arise. So the constraints will be refined and sharpened to also be more stringent on the physiology of the higher order layers.
But now you don't have a multi-layered representation of the cortical areas, right? You don't have a 6-layered cortex. So
The model can be easily 6-layered. So yeah, in that sense, it's already deep. Whether it's cortical, depends on your demands. I wouldn't say it's cortical in that we are yet unsure about what happens over here. So whether that contains, if we train a network, for instance, on boats and cars and objects, that we have cells that would be specifically sensitive to the class of boats, that they always respond to boats regardless of viewing angle. And translational properties, etc. So in that sense, there's a lot of work to be done. But I find it, a sort of instructive and a very concrete approach to work on the notion of world representations. I'm going to switch here to multisensory interactions, to physiology, because that's what we also do in the lab, and ask, well, if we simply look at a creature like the mouse, and we look at its visual cortex, isn't it indeed the case that there are multisensory processes going on? Is there, for instance, sensitivity to audition? The technical approach is illustrated here with a little movie of Guido Meijer. It's 2-photon imaging in the mouse visual cortex, where you see how a genetically coded calcium dye, GCAM6, reports fluorescence. And you see upon this grating that will move, cells appear that light up, so to say. Yeah, there we go again. They light up because their somatic calcium concentration rises, and that's due to firing of at the level of the soma. There's some red cells, which are the somatostatin positive cells. That's not of particular interest in what I will present, but these are all the possibilities of genetic engineering in that sense. And you heard some of it yesterday in Edward Mozart's talk. What we see here is an image of the mouse visual cortex with 4 cells highlighted. Their calcium traces or their fluorescence traces are plotted over here. It's a long time scale with the change in fluorescence relative to the baseline plotted. All the colored bars are presentations of oriented gratings that move. And in some cases, we add this auditory stimulus at the crossover here. It's quite usual to find orientation selectivity, so cells are more responsive to certain orientations of the gratings than others, as in this polar plot over here. The question, of course, is then not to get the usual orientation tuning curves, but to see what happens with auditory stimuli. And they indeed appear to modify the tuning curves. So this is orientation of the grating. This is the delta F over F response. Purple is visual only, and blue is the auditory stimulus added. And what you see are sometimes suppressions in some cells. So this would be a single cell with multiple repetitions of the stimuli. Sometimes also enhancements by the addition of an auditory stimulus, a frequency modulated tone, sometimes a shift in the curve. Overall, you could say, well, these are up and downward changes. There's no net suppression or enhancement. But it turns out to be the case that if you plot all the response changes according to an index of change with the neuron count, you get a broad distribution. So there are indeed both positive, let's say, excitatory and inhibitory or suppressive changes. And they're broader than expected by chance, which is borne out by the fact that you can compare this distribution and its width to a shuffled distribution, and then the true width of the curve is exceeding what you expect by chance.
Can you explain how you add the audio on the visual?
Yeah.
How is the method here?
Well, the mouse is awake in this 2-photon situation. It's head fixed. It's watching the oriented gratings, and then the frequency modulated tones are being played or not. And it's interesting, I don't present it over here, but there's also a story here how congruency between the tone and the visual pattern matters for the enhancement or suppression that you get. Yeah, that does matter. It also matters whether you take a frequency modulated tone, which has a rhythm to it, versus white noise, for instance. That also matters. White noise tends to give more suppressions.
Cyriel, how do you exclude that this is not a non-specific attentional modulation?
We look for that at the eye movements and pupil size, which is in many ways a measure of arousal, not totally comprehensive, I would say. But it turns out that the visual stimuli give a similar kind of pupil size change as when audition is combined with visual. So that would argue it's not purely an arousal effect. But one could maintain, you need to do more. You might monitor heart rate in addition and other measures. Of course, the problem that whole set might not be inclusive about all arousal processes.
I hope I'm not anticipating something you're about to say, but I think one way we can observe this sort of multimodal integration as as people is that the McGurk effect, and where you watch someone's lips and it changes the sound you hear.
Yes. Yeah.
And I was wondering if if there's any possibility of doing something, because you mentioned something about the congruency of the
Yes. Yeah.
auditory and the visual stimuli, and I guess with the mouse it's less easy than maybe for certainly with humans.
Well, to have
you have some ideas of how to really measure the impact.
Psychophysically, you mean? Yeah, it's been done also by work in auditory cortex of Christoph Kaiser, for instance, and Charlie Schroeder did work on it. Yeah, so it's McGurk is hard to do, as you say, in mice. It's a very strong effect in people where, for instance, there can be sort of a binary perceptual decision whether you hear a ba or a da, depending on the lip movements that you see. So in that case, audition has a cross-modal influence from vision. Here, we don't look at perceptual decision-making per se in this experiment. We do have audiovisual detection experiments going on, so I hope to have that ready in some time. What clearly matters at the level of these single-cell and population responses is the congruency indeed. So if it is the case that your oriented grating is moving with it has a spatial frequency, but also a temporal frequency. If that is congruent with the auditory frequency modulation, you will see this type of heterogeneous pattern. If they are incongruent, so let's say if there's a fast visual moving grating together with a slow auditory tone modulation, that kind of incongruency is less likely to lead to excitations or stimulating effects on the visual responses. And then white noise, more unstructured auditory input, would also tend to suppress the population more than shown here. So it is quite important. It might have to do with rhythmicity that is induced either by way of audition or vision, and which could be imposed through cortical-cortical connections on the other modality. Another question, for instance, is whether there's purely tone responses in the visual cortex. So without any visual input, do you get responses to sounds alone? This is also the case. It's not hugely distributed in the population. There's about 6.3%, but those neurons are significant. They are occurring more than random. It's the delta F over F response you see across time with the auditory stimulus over here. And you do have these single cell responses. They tend to be more laterally localized in mouse C1 if those cells are indeed auditory responsive without visual stimulus. On the medial side, there's less of this auditory responsiveness. Yeah. In this case, there was no visual stimulus. So, yeah.
What would be the hypothetical relation between that sound stimulus and the visual stimulus these neurons provide? Any?
We don't. This is purely to test for auditory responsiveness without visual input. The previous slide is about when you stimulate them together, and there the congruency matters. Yeah. We don't know yet whether this relates to some kind of developmental plasticity, where you could think of animals locomoting, and if they locomote at a high speed, then the speed of visual change is large in the optic flow. Plus, the rustling of leaves, etc., will also occur at a higher frequency. So there are these kind of ecological congruencies which make sense, but we don't know how it arises exactly.
Like you present the stimulus for 2.5 or 3 seconds. It's a very long stimulus.
Yeah. Yeah.
Right? So you have these really long-lasting responses now. And I guess you need that for your optical imaging.
Yeah, it's also to sample. Yeah.
If we would move in a more ecological range of sounds that you usually use also in your tasks, like a more transient sound, would you expect these signals to be equally slow? Do you expect these slow responses? So would there be anything different in the response to auditory stimulus in terms of the dynamics as compared to the response to visual stimuli?
We haven't tested that. We know that there are P1 neurons that prefer slow stimuli, slow gratings, and other would prefer fast-moving gratings. That translates to differential sensitivity in the higher visual areas, AL and LM, as shown by Mark Andermans, for instance. Yeah, we don't know much about the ecologically more valid stimuli, such as what the animal would hear if it would run fast versus slow.
And what intensity is the sound at?
Well, that relates very much to the background of the two photon scanner. So it has to be clearly a number of decibels above it. I think we take 20 decibels higher, which is clearly audible. And then the age of the mice is important because they tend to become deaf after several months.
So you have a mouse with a headache responding to this.
With a headache.
Yeah, because the sound will be really loud.
Yeah, but it's, I mean, it's transient. They can deal with it. They don't startle or, yeah, have
No, so 2.5 seconds.
pain-like behavior.
What, 80
Yeah.
decibel noise or?
I wouldn't say by head. I would have to look up it in the paper how loud it was. Yeah, but you do have to get clearly above the background of the.
But you're driving the system really strongly, right? It's not like subtle. It's not a subtle manipulation.
What Guido did was, in fact, to apply lower contrast in vision and some lesser intensity on the sound. The pattern of responses does stay the same then. Yeah, okay. It doesn't seem to be critical. Yeah. All right, yeah, Diogo.
Still on the previous slide. I'm sorry,
The previous one, yeah, sure.
yeah, I'm too slow. Like, do you see any kind of learning happening here in terms of like early trials compared to late ones in the response of tone and visual associated stimulation?
Not here, because this was just a passive situation. The animals just listen, and we do have the other data now on an active audiovisual detection task, where you do see learning. That is, animals become better in detection over time.
So would you expect it to be some sort of top-down prediction of the auditory ending up at the visual?
Yeah, it could be. It's sort of interesting to consider whether auditory is top-down or sort of equal level interaction, lateral, so to say. But it's very well imaginable that these connections are facilitated. We see in the psychophysical experiments where you do a, let's say, visual contrast on the x-axis and, let's say, detection behavior on the y-axis, you see a big shift when an auditory stimulus is added as a sort of aid to the visual detection. And this is happening in a Bayesian optimal fashion, but it also improves across training. So it's actually a bit better than Bayesian, it seems. I shouldn't say it because it's sort of hard to defend, but there are learning effects. That's what it comes down to, yeah. All right, let's move on. So we're going to look now at a higher stage of supposedly multisensory processing, the perirhinal cortex. Because when we talk to anatomists like Menno Witter, who also works with Edward Moser, he says, well, if you want to look, for instance, at integration of audio and vision, please look over there. It's a structure for recognition, for object recognition, particularly implied, and also tactile and visual information comes together there in particular areas. So we said, well, let's do that, but the results turned out to be quite different than expected. The electrophysiological setup is over here. It's a quite complicated recording paradigm where we record ensembles in 4 areas at the same time. This is work by Martin Fink and Jeroen Bos. That is barrel field for tactile input, so somatosensory, area CA1 of the hippocampus, V1, the visual cortex, primary, but now in the rat, and perirhinal cortex deep down here. So it's close to the entorhinal cortex, but classically implied in the WAT pathway of the medial temporal lobe that is more object-related processing. Whereas, for instance, medial entorhinal, as Edward Moser explained, is much more concerned with space, we think. And here are some of the tetrode bundles which can be directed to these various areas at the same time. And the task was initially set up to be a multisensory task. In the end, it was quite difficult to do for the animals, and we ended up doing a visual discrimination task. The rats were positioned on this figure 8 maze. Based on these visual inputs, they would have to choose whether to go left or right. Decision depends on where the CS plus is. Let's say if the CS plus was the diamonds, they would have to turn left and follow the money, so to speak. ignore the airplane. After that decision, they would have to pass along this patch of sandpaper as additional somatosensory cue, and then they pass a point of no return over here. They cannot go back after having passed that particular point. They reach a reward site, return to the middle, get maybe an additional reward, and have to wait for the next trial to arrive. So it's quite a difficult task to learn because of this visual requirements, and the visual stimuli are also isoluminant, so they really have to look at the pattern. But there's no strict multisensory requirements yet in this task. For the results, it doesn't matter so much. You do find in the perirhinal cortex a lot of cue responses, let's say to an auditory beep that cues that the image will be forthcoming, or to the visual images. But the most striking pattern in about 70% of the cells is that they have these sustained firing patterns. And you see this when you plot the spike rasters as a function of passing the point of no return here at 0. You see how the red trials show a firing rate enhancement, which is for one particular arm, the left arm, whereas the green trials are for the right arm. There's no firing rate enhancement over there. So it's highly differential. The cells either are very active in one arm or have no change in the other arm in this case. And there are variants on this pattern. This second example would say, well, in one of the arms the firing rate goes down, and in the other arm it stays more or less the same, which is also different because the perirhinal cells have a high baseline of firing. So you can actually go down, unlike place cells in the CA1 area, for instance. They're basically quiet except for the area of the place field. Those mark the left versus right arm trials. So red would be the leftward and green rightward. So for CA1, you see in time, so these are two adjacent peristimulus time histograms with point return to the middle over here. For CA1, you see punctate responses as you would expect for a normal place cell. That is, the animal, for instance, takes the left arm again, runs through the place field. This gives a burst of firing over here, and not so in the other arm because there's no place field over there. So it's much more punctate and brief. Also in the barrel field, you find transient responses, for instance, when touching the sandpaper, but there's no systematic sustained response. And spatially, this leads to this remarkable pattern of very large coding segments that are differentially expressed in the perirhinal cortex. So there are perirhinal cells with, let's say, all enhanced firing in the left arm, but then throughout, or both enhanced on the left but suppressed on the right, as over here, or sometimes a mix or this enhancement in the middle. So there seems to be much more coarse coding going on that at least has all the looks of spatiality to it. Little to do with object coding unless we say, well, also an arm is a kind of very big object. It's a kind of unitized entity that the rat knows and must know to perform its task correctly. So when you plot all the units in the spatial domain again, you get the following picture where the perirhinal cells are over here. So now we have up to about 350 units, CA1 cells down here, also about 350 units. In color, you see the rate difference between the preferred and non-preferred arm, the preferred arm being the one with the highest firing. And you see these yellow bands, these stripes that are intermitted by these red responses, the peak responses, by which also in CA1 you see a tessellation of the whole structure, the whole task. That is normal. But you also see how when you break down this rate difference between left and right, the rate differences consist both of enhancements, as expressed here as the yellow stripes again and red stripes for the preferred arm, and the suppressions in the non-preferred arm. So you also see here that relative to the spatial bins that belong to the baseline or the middle patch usually, there is clearly a suppression in a lot of cells. So it's a combination of enhancements and suppressions that you find, which is different from CA1 again, because there, the rate difference is made up of the place cell responses only. There is no active suppression component there. So two differences. CA1 is much more punctate as expected, But also the pyramidal has a clear suppressive component, which has also been somewhat described in vitro physiology.
If you now would do a decoding experiment where you would predict where the animal is, given the neural response, which of the two would you think be more accurate? Giving you more precise information about the task segment that you're in.
Well, if you have enough cells in your population, the CA1 would be clearly more accurate if you want to know exactly where you are. But maybe CA1 is not always available for you to help. And it could be, for instance, in procedural tasks, quite handy. Let's say if you're a cab driver or so, to know in which neighborhood you are, whereas you don't need the detailed knowledge at the same moment. So you know, when you have to go from, let's say, Sagrada Familia to the airport, you just worry about the big routes, and you're not going to worry about which house of Gaudi was built on which particular street, which is more the detailed place-like knowledge. You just worry about, I'm in this neighborhood, Exemple or whatever, Gothic Quarter, and that's what matters at the moment. So it's coarse knowledge, but it can be very useful not to involve your CA1 all with the full intensity always. So in that sense, yeah, it can be a coarse build-up stage towards CA1, but in many cases it might suffice for doing your task. All right?
The speed along the task. So basically,
Mhm.
cells tend to fire in the portion between the point of no return
Mm-hmm.
to the central. For that portion in particular is where you get most of cell firing, both in perirenal and, I mean, from light blue to.
Mm-hmm.
So how is speed and how is the behavior of the animal in the task? It seems to be that most of theta coupling is occurring in that portion where the animal is just running
Yeah.
to get
Yeah.
quickly into the decision point again so that
Mhm.
start again.
Yeah, yeah. So running speed is low here, generally because the animal will have to wait for the next stimulus. That's a kind of variable period. Here the running speed is quite high up to the reward site. After that, behavior could be a bit variable, but usually the animals run towards the middle lane again.
So this basically means that the amount of time that is contributing to your statistics along the different portions is different. So you are
Mhm.
mapping different time temporal frameworks into the space.
Yeah, yeah, that's correct. That's also why we like to have many trials to be able to compensate for the, let's say, the speedily visited locations. But also, let's go back to this slide where this is all temporal scale, and you also have the sustained pattern. So also in the time domain, it's a very consistent pattern of sustained. This one.
Aligned by the point of no return, which is there.
Yeah, yeah. Many cells will also start firing here already at the branch point. But that's very close in time. That's true, yeah.
Do you think it's significant that the peak density of coding, if you go to the next slide, for CA1 is more towards the center of that turn as compared to the peririnal? So is that significant to you?
Hard to say. It indeed correlates with theta, as you say, because in the middle the speed will be lower and the behavior is a bit more variable because the animal is waiting a bit for the next stimulus. And because theta and speed are lower, the firing will tend to be less discrete and the rate will be lower overall. So I don't want to say too much about this middle segment, except to say that the peririnal cortex does show in a subset of cells discrete coding for that middle segment, even though behavior is highly variable. Of course, one could get a load of critique if you say that the peririnal is doing something spatial, because classically it belongs to the WOT pathway, that is object coding, and that's where a lot of the findings go. Let me say first that there is indeed a link between the peririnal firing and theta rhythm in the hippocampus, because the more spatially discriminating cells also tend to lock more to the hippocampal theta rhythm. And you could view these, let's say, neighborhood-like fields as being driven from the bottom up, that is, as a sensory bottom-up integration, but they could also arise by integrating multiple place field-like inputs from the hippocampus in a top-down fashion. And the theta locking suggests that there is there is indeed an intimate relationship with the hippocampus. For the rest, we don't know very much about the mechanisms. Anatomically, there is a direct perirhinal to hippocampal projection, so it doesn't have to go all through the entorhinal cortex. Now for the spatial matter, we sorted out a number of questions, trying to see, for instance, if it does matter if the animal makes a correct trial or not, an error with no reward. Well, does it matter for the sustained firing? It does not matter at all, actually. You don't see any change when the animal makes an error, but still completes its run. We looked at a variant of the task where the working memory load was varied, in that the animal does get the stimulus, but it cannot move directly and make its run and decision. It has to wait a little bit. Also did not matter for whether you had the sustained firing pattern. The nature of the visual configuration or the sandpaper configuration also did not matter. It was not stimulus dependent in that sense, whether there would be sustained firing on the right or the left. Then there's the matter of direction of rotation and maybe acute environmental cues that could say basically, well, you're here on the left because you are turning left if you make a left turn. It is clearly a vestibular signal that might be sustained across, let's say, the whole left arch. We tackle this, also the problem that, yeah, in particular, spatial views of the environment, there could be some kind of object being detected, like a Neuralinks rig, for instance. But if you study the turnarounds of the animal, in particular trials, this occurs. For instance, the animal goes to the reward side and doesn't like to go on, it just moves back, it walks the other way. Then you see, so with a distinct, different turning direction, that there's still, at least globally, a good correlation with the forward locomotions. The spatial selectivity of the cells is retained, although there's going to be dependence on locomotion speed and what the animal precisely is doing. So in that sense, we have indications that indeed these cue and rotational directions are not directing the difference between left and right. Yeah.
Question. So taking your example before of going from here to Sagrada Familia or to the airport, so a large scale path going on. So back then, the modeling perspective of place cells would be like, if you have to actually now decode a large trajectory, you might use, large scale place fields so that, it's not that expensive to compute for sequencing and so on, right? Yeah. But now you seem to have a better answer for that. That is, instead of getting each place field, I'm gonna compute every single neighborhood that is in between my current location and the target, which might be really cheaper computationally from a computational perspective, right? Yeah. So how much of those phenomenon that we find in place cells, such as sequencing, would you expect to have in your neighborhood cells? So would you know what I mean? Like if you now have multiple neighborhoods, would you?
Yeah, well, we haven't studied this in still larger environments. We do look at replay, of course. We are a big fan of sleep replay and other research. So what we do is, because we have these hippocampal sequences, we can compute hippocampal trajectories and see which ones come back during sleep, and then see if the perirhinal responses correlate to that. Let's say, are they pointing to the same left or right side? I should mention the work of Kjellstrup from the Moser group that is on ventral hippocampus. They recorded very large place fields also, if in these rats walking huge tracks, which is beautiful. However, if you compare it, at least the dorsal hippocampal cells with the perirhinal here, and we had the impression that the hippocampal cells, the place fields are just scattered across the whole environment, that there's little system. Whereas the perirhinal cells, in their onset and offset of the firing field, seem to be locked to the branch points, point of no return. So we computed that, the onset and offsets of the place fields of CA1, and indeed they are more scattered. There's a much bigger variance. So in that sense, we could say the perirhinal cells are more locked to the geometrically and behaviorally important markers of the task, which could indicate that these neighborhood fields are also more task-related, maybe dependent on prefrontal input, which is quite strong to the perirhinal cortex. But that is all to be tested. It's basically speculation. It is, there is a difference to the CA1 cells in the locking to branch points and points of no return. Okay, let me, if this is okay with you, go on to the last part, which is got to do with the hippocampal function. We're quite far from consciousness now, although it might be related. So here the question is, well, you know about place fields, other correlates of hippocampal cell behavior, but it all relates to, of course, the animal itself. Let's say it's ego-bound because it's about the animal's own position. And the question is, following up on the mirror neurons, does CA1 maybe code something more about other beings? And the experiment we designed with Paul Verschuren here and other people and executed by Martin Fink and Jeroen Bos again was actually to have a rat walk on a maze over here. But then the rat would be transferred to this observation cage over here, while there would be an epoch, a little robot moving about, remotely controlled on that maze. And the question is, do the hippocampal cells signal something about the location of the robot or its behavior? And I have the little movie of that. This is really, in a way, old times, because the project was started, I don't know, 6 or 7 years ago or so. There's a lot of complicated analysis steps. But here you see the rat with the head stage in the observation cage. Depending on where the robot is moving, it can get a reward by looking into a left or right well in the cage over here. You see all kinds of events being reported here on screen. Now the robot is moving from here to this middle start position, and it's performing the so-called front task now, because that front task is just meaning that the robot moves in the front lane to end up here again and moves in the front lane. And then the rat would see, okay, the robot is now on the right, so I can gain a response by licking on the right at some point. So there's an element of observation of this other being. Of course, it's quite handy that we can remotely control it. A real rat would be totally erratic in its behavior and might not be locomoting at all, so that's always complicated. The whole experiment looks like this, so it has a lot of control phases in it. There's a phase where the rat itself is moving on the maze. Also at the end, there's maze 2. Here's the front task, here's the mid task, where the rat is in the observation cage. And there's a phase of where the rat is in the observation cage, and the robot is just freely moving without any task requirement. There's also a period where there is no robot at all. Note that the behavioral requirements are the same for the front and the mid task. What matters is whether this robot is moving just in the front lanes or down here into the middle. And it's crucial to have both the firing rate plotted as a function of rat position, which is in the upper row, or as a function of robot position, which is in the lower row. So these are the robot positions for this middle task, and here with the free roaming, it just moves about. First question is, are there mirror place cells? Disappointing answer might be no. We don't see them. You have a clear place field here of the rat on the maze, as you see, and it has firing rate. And that's both in the first and the second maze period, so at the beginning and the end. Blue is all the recorded head positions of the rat. If you then go to the period where the rat is in the observation cage, and you look at the plotted robot positions, in there's no enhanced firing. You barely see any spikes in this case, probably because the rat's place field is over there, and the rat is actually in the observation cage. So it's at the wrong place to fire, so to say. But note that there is overlap. If the robot would trigger in its position firing, it should have been visible over there. These are just control plots for the front and mid task, robot position, firing rate map, and the controls. We had one occasion where we did record a clear place field for the robot at the same spot where the rat had its place field. And there it was quite important to reanalyze the data, go back to the video, because actually, as you might see over here, there's quite a high door here, plexiglass, but the rat actually climbed over it and went on the maze, and which explained our robot place field, so to say. So always go back to your original data if you can. There are, however, also some positive results to mention, which are also striking. You see, for instance, a cell over here that has a distinction between the mid and the front task. This is the firing rate by red position. This is the firing rate by robot position. You see already that for the red position, there's an enhancement of the firing for the front task. And that translates into an overall higher firing rate for the whole front task as the robot is doing that. But pretty much all positions. There are some cells that would distinguish a little bit by position, but quite coarsely, so it's not something we would emphasize. And this can be more detailed in the sense that below here we have a plot that shows, well, for the, let's say, the inward movements. So when the robot is moving towards the center, the firing is enhanced in the front task, but also in the mid task, so in the other task variant. Whereas when the robot is moving outward, the cells are not firing very much in both cases. It's true that this also could relate a little bit to reward expectancy, but you basically see the firing rate difference across the whole stretch, for both tasks. So there are interesting modulatory patterns. It's more like an overall rate coding scheme than a very discrete coding of what the robot does or where it is specifically. The overall behavior does tend to show differences. And now that these kind of differences are not explained by different rat behavior. That's always the plague, that the rat do something different in case of one task condition or the other. All right, so at least except for the discussion, I come to conclusions. We've seen all the way from consciousness, like I said, it's an impossible thing to do, but all the way from consciousness where we see the emphasis on multimodal surveys, multisensory interactions, eventually in the service of goal-directed behavior, how we moved on to predictive coding, the initial steps towards world modeling in multi-layer structures. We do have V1-like responses over there. We don't have a clear sight on how the higher order areas can be trained to generate also semantic or other higher order representations, but that's a topic to be done. These networks are trained on biological principles, so not backpropagation. In relation to this, we see now the visual cortex is already in its primary stage more multisensory than we thought. It has this auditory influence, both excitatory and suppressive. Paranal cortex, a bit unexpected, much more of spatial coding, we think, than stimulus-specific coding. It's also there. I didn't emphasize it, but there are these item-specific responses. So it turns out to be more of a mix, but there's also a lot of work to be done to sort out what this chunking is about, how it's generated. And then the part about self versus others, that there are sensitivities in the hippocampus to behaviors of strange other beings. Let me thank a number of people in particular. Here is Jorrit Montijn, Guido Meijer, Karin Dansing. They worked on the 2 photon imaging parts. Shirin Dora and Sander Boté at the Center of Mathematics worked on the modeling, the predictive coding, and Martin Vink and Jeroen Bos and Laura were instrumental for the paranal cortex as well as the hippocampus. And then there are, of course, our external collaborators like Paul and others to thank. So thanks for your attention and questions already. Yeah, I'd like to leave it here. All right, other questions for Cyriel? Yeah, Lisa.
So regarding this possibility of the hippocampus representing the space that is being occupied by others, either robots or rats, did you ever try a rat instead of a robot?
No, we didn't. Some other people did actually, like Heimendahl et al. They tested whether there would be cells, a bit like the Jennifer Aniston cells, that would respond to specific rat individuals. So let's say rat A or rat X. I don't think they found very much there. They did have a paradigm where the presence of a rat would matter, I think, to some cells. But they didn't test for the behavioral patterns.
So because basically, if I mean, the hippocampus, the role of the hippocampus is just to try to get a record of those contingencies and eventually related with the needs of the animal. So I guess that if the animal, for instance, see another animal getting fed, for instance, on some place of the space in which he already has a representation, I guess that should be signaled somehow to recognize. So imagine you just feed the animal at some point, like the goal direction task, and then the animal see other trying to go there. That should trigger the hippocampal representation in a way.
Yeah.
More than a robot. I mean, at the end,
Yeah.
the robot is coming close, the animal feel it as an amenage because something is coming close.
Yeah.
And then when the robot is far away, then those decrease. But basically it doesn't generate any problem or conflict for the animal because he will get fed anyway. But
Well, yeah, the robot here is relevant, of course. It's directing where the rat should make its licking response, even though it's a tiny response. Yeah, there are complications. I see what you mean, that in a natural situation, it would be very important to understand for a rat where another rat is, and especially if this is a feeding location, you get questions of competition, aggression, and maybe collaboration, cooperation. Yeah, whether that's all coded in the hippocampus, I don't know. Once you work with other animals that do significant things, you also have to deal with the rat's own behavior, even behavioral tendencies to go there. So there we get into planning and action preparation and motor readiness and stuff. So it does become quite easily confounded if the coding of others becomes too significant.
yeah, but what I found important is in the design of the task to try to incorporate the natural influences that the animal should be confronting with. So that, I mean, those circuits has been developed for solving those contingencies. So I guess that...
Yeah, very right. I think, yeah, there would be a fair chance to find something in the hippocampus if it was worth remembering to be stored in episodic memory. There could also be very well prefrontal circuits and, of course, amygdala that are triggered, so probably a whole network. Yeah.
I just wanted to say, yes, so in amygdala you see neurons which respond to individual other rats. So a given rat will have a given neuronal response specific to it, and it will endure actually after the rat has left. So the amygdala may provide the memory for this kind of thing.
Okay.
A single unit response.
Right. Is this dependent on the, let's say, the emotional value of this other rat? Whether it's
well, no, in the sense that I think this is not published yet, but O'Keefe put sort of female and male rats with the other rat, and it didn't matter. There were neurons that responded to individuals. It didn't seem to
discriminate between gender.
It wasn't obviously related to value, but we don't know what the value is. But
okay, yeah.
And in human epilepsy patients, you know, you see these Jennifer-Aniston cells. They're actually slightly denser in the amygdala than the hippocampus, and they're also their cells that respond to spiders or, you know, individual objects. So it may be that these kinds of specific items are represented more in the amygdala than hippocampus.
And
interesting.
Also very recent data suggests that CA2 cells, they have CA2 cells, which for so long has been ignored as a transition region. Actually, what they have been signaling is this social interaction, and at the dorsal hippocampus reflecting that contingencies between social aspect of a space. So that probably, and the way in which these CA2 cells connect with CA1 cells is pretty much specific. So
it's mostly social recognition memory. It's quite different from, let's say, the online coding of what others do. But I would agree, CA2 is important.
But now maybe if you look at the next slide, when you do this behavioral analysis, in the lower part, you were talking about it's the movement of the robot that is modulating the hippocampal response. But then we can of course wonder whether that is a movement that is interpreted by the rat in egocentric terms, like something is coming, an agent is coming towards me, right? Or whether it's really a spatial representation, like some object moves in space. And I think this is I think a distinguishing question that we can now pose here. So what's the answer?
That could be also the case, that coming towards you is more of a looming signal that.
When they come towards you,
Yeah,
he's like,
I know.
oh, Paul is an X, Y, 1 and 2, or am I going to threaten you, right?
All too familiar with that. However, where is the slide? Oh yeah, like over here, you see that there's an overlapping portion in space where the robot is both in the front and the mid task. That is this middle portion that we call it move back or move from the start position overlaps, and you still see that given the same spatial position and more or less the same distance, there's no firing here, but there's firing over there. So it would suggest that there's something about the robot's overall behavioral pattern that matters, and not only the imminent acute cue position.
No, but then the question is this like an inference on, let's say, the movement vector of the other agent and the possible consequences behaviorally, and then maybe triggering some valence response, like a threat, right? Or is it just saying like, oh, this object is now an XY1 and now it moves to XY2?
Well, again, the behavioral requirements and I think also emotional expectations are the same in the front and mid task, because the behavior of the rat, what he has to do is the same, the reward expectations are roughly the same.
Yeah, the rat feels safe there. I mean,
Yeah, but
he
in
has learned over the periods that there is nothing else, that something coming. So
yeah.
And the rat also knows that the humans engineered the box small enough that it can still escape.
Yeah. Well, we did our best, you know, but notice that also when there's no firing, you could say, well, there's firing all the way over here. It's not particularly vectorial. But here's also a moment where the robot is really close to the rat. So in that sense, if it were a threat or some approach-dependent response, that wouldn't hold here. It's a bit different over here with inward versus outward movement, as there might be.
anyway, Mari Sanchez Vives has tested this,
Mm-hmm.
and they have been recording some spatial. I don't know if you know this.
Yeah, very familiar. Yeah. Okay, thanks. Yeah.
Please, go ahead.
Yeah. Did you check how the rats react to the robot if they find it stressful, its presence or something? Like if they were in the same cage, you know, as the robot is moving around, do they find it stressful or what?
Well, we did these pilot tests indeed to see how the robot would deal with the rat would deal with the robot directly, and first, upon a direct confrontation, they might be a little bit scared and hesitant. They hold back. These robots also can make sounds like funny sounds like oomph or something, and they have flashlights. You know, these are from the epochs of GTEC, but in the end they get annoyed, and if the robot has no consequence, you know, we even had a rat that threw over the robot, and the robot couldn't move anymore. So they become a little bit arrogant towards it. Yeah.
So while the rats were watching the robots, were they did they have any reward at all?
The rats could get a reward at one point. This is when the robot had completed the cycle. The rat could get a reward by poking in a left or right side.
They didn't get rewards in specific locations.
Yeah, these are locations right next to each other. Basically, these two boxes in the observation cage are the reward sites. So we don't see a particular place field differentiation over there, actually. You do find sometimes like, do I have a slide of that? Like over here, you do find, let's say, a place field in the observation cage, which then translates into this robot position picture. There we don't see very much differentiation between the left and right reward side. They're simply very close together, and we don't think the cells discriminate. Yeah.
So one last question from my side. So Laurent Frank has shown that CA1 is kind of suppressed when the animal is stationary.
Mm-hmm.
And that also leads to the question of what's happening with the theta oscillation in CA1 while the animal is actually in the cage, compared to when it's actually exploring. If that would be actually a reason why cells would not be potentiated or, you know, triggered sufficiently strong to reactivate when the robot is. in
Yeah,
terms of-
yeah. The theta rhythm is a complicated rhythm. Originally, also people like Buzaki and so described how theta can be elicited by attention and orienting responses. So it's not only locomotion. Yeah, these are salient sensory conditions. So we do also see theta while the rat is in there. And there are periods where it will leave its reward sites and just move about a bit. These are, giving rise to deviant head directions, and in histograms like these are cut out, so we focus on the observation orientation. So yeah, overall, we looked at the theta. We looked at whether there was a difference in the theta phase locking, for instance. But theta is variable. there is some, but not all the time. Not as during locomotion. Yeah.
What happens when the novelty of the robot wears off?
The novelty as the task is going on. Oh yeah. Rats go through several sessions. We often cannot say for sure whether the same cells are recorded from one session to the next. We don't have the impression that the responses overall decline as animals enter into repeated sessions. It might have to do that they're simply motivated to watch the robot and do the task. You do find a bit less correlates to the phases where the robot is not of interest. So this free roaming period, for instance. Basically, the rat is left on its own and will generate its normal place fields, but only for the observation cage. So,
Right. I guess the success rate for the selection of the nose poke site is relatively high for the rat while it's observing the robot.
the success rate, they're not terribly good at it. You would say it's a very easy task, but overall they score between 60 and 90% correct across rats. So they do make quite a bit of errors, yeah. They might have the idea, well, I get my reward anyway somehow. So
Potentially that might explain why perhaps the rat does not exactly position itself in the place of the robot when it's reached.
Well, it would be very interesting to do another experiment where, like Lisette, where the position of the robot does become precisely significant for the consequences of the rat. Not as in this case, the overall left or rightward movement. So I think there's room for follow-up experiments here. Yeah.
It only took us 6 years to get to this point, right? So
yeah.
we'll run another experiment.
We'll be retired then, but it doesn't matter.
All right, well,
Yeah.
last question.
Yeah, it was more just really quick comment, because you were saying with regard to this error rate, I think rats tend to make quite have a sort of pretty high sort of minimum error rate, right? I just remember Matthew Diamond saying it was like 15% or something, even in a fairly easy task that they designed, which the rat had a lot of time to learn. So
15% failures to.
yeah,
Yeah,
it was like a sensory discrimination task, and the rats had it, and he interpreted Matthew Diamond as some like they're built for more variable environments, so they shouldn't be learning these rules to such a high extent.
Too fixed, yeah. That might be the case, yeah. Here, yeah, it is a visual task. They, unless the visual cues are very clear and salient, then it would be easy, I think. But here the robots tend to be remote sometimes, and the rats really have to pay attention, so they might be distracted every now and then, yeah. But I agree, it's hard to get them really systematically above 90% on these kind of tasks. Yeah. It's also not so bad because the error trials are also very informative, like you saw in the Paranal task. Yeah.
So to finish up, how does this task inform your theory of consciousness?
Well, I already indicated it was a long stretch to make.
Yeah, but I rejected that. So
yeah. Well, we haven't talked very much about, well, I talked a little bit about the semantic interpretation of scenes, so that usually part of a full conscious representation would be an interpretation of the scene, like seeing you would, sort of evoke the concept of Paul, that you're there, and so for Jennifer Aniston of Obama. So yeah, part of this this book actually, which this book is a must read. Anyway, very good. Is a section actually on the interaction between semantic memory and perception. That is to say, that for a full conscious representation, you would normally like to have sensory input interpreted as something purely visual with a semantic connotation attached, so that you know the picture of the duck and the rabbit, where you can switch. Wittgenstein used it, and it originally came from Jastrow, but it's often called a perceptual switch, but it's actually switching between duck and rabbit, whereas the sensory input stays the same. Mm-hmm. So that's where I think hippocampus anterior medial lobe is very important for giving you a full representation.
All right.
Yeah.
Those words.
Okay.
Thank you very much, Cyriel.